Welcome back to our practical class. In this video, we shall be dealing with base composition analysis of a DNA sequence using Python. Understanding base composition is a fundamental step in bioinformatics and sequence analysis. And when we talk about base composition, it refers to the proportion or percentage of each nucleotide base present in a DNA sequence. The DNA consists of 4 nitrogenous bases, which are the adenine, thymine, cytosine, and guanine. So base composition has to do with how many of each base, that is the ATCG, are present in a DNA sequence. So for example, if you have a DNA sequence just like the one on the screen, how many adenine are found in this sequence? How many thymine are found there? How many cytosine? How many guanine? That is what base composition is all about. And we have a formula for calculating base composition. The formula is base count divided by total length times 100. So to get the base composition of adenine in a particular sequence, you count the number of adenine, then divide by the total length of the DNA sequence times 100. One may ask, why do we calculate base composition? The calculation of base composition in bioinformatics helps scientists in genome analysis. Even in species comparison, scientists can compare the base composition of a particular species with another species. It also helps in identifying gene-rich regions. When you analyze the DNA sequence, area with a good number of base composition signify a coding region. Then it can also be used in evolutionary studies. Also, it helps in quality control in sequencing data. These are the reasons why we calculate base composition of a DNA sequence. Now, let's move to a practical session where we are going to use Python to calculate the base composition of a particular DNA sequence. This is my desktop, so let me open Python. You type Python, this is it. Python has opened. You can see I've already written the code because of time factor, and I use comment to explain each line. So the first line is where I define the DNA sequence. That is where we have the DNA is equals to, then we have our DNA sequence. Then the second line is when I told Python to use uppercase, that is capital letter. Then the next line is total is equals to the length of the DNA. Then the next line is where I told Python to count each of the base in our DNA sequence. That is where we have DNA.count A, T, G, and C. Then the next line is where we have the print statement, where we are telling the Python to give us the result. Then we told Python before printing out the result, it should add, that is, it should check the total number of each base divided by the total number of the DNA sequence times 100. That is the formula for calculating base composition. So we said Python should apply that formula for each of the base and then print out the base. As you can see, once I click enter key, the result is there. You can see the final result is out. Sequence, our sequence has been shown. Then you can see the length of the sequence 13. You can see the number of A in the sequence 3, that is 23%. T is 4, 30%. G is 3, 23%. C is 3, 23%. So this is how to calculate DNA based composition using Python.